Hey Team Bio, welcome to your screencast on Mitosis Explained. So, today in class, we organized this mitosis puzzle, and you started to be introduced to the different phases, their structures, and a little bit of what's happening in each phase. But just to briefly relate this image to what we saw on the screencast yesterday, I told you that the cell cycle can be broken up into two major components. First, or I guess that was at the end of the last screencast, we had mitosis. And then we had interphase. And so interphase proceeded to mitosis and cytokinesis, where after mitosis and cytokinesis, you have two cells, and then you enter back into interphase, and the whole thing loops around again. And we said within interphase, there are three different parts of interphase. We've got the G1 phase, followed by the S phase, followed by the G2 phase. And the G1 was growth or gap 1, where the cell is growing and doing normal cell life stuff. And the S phase, the cell is also growing and doing normal cell life stuff, but on top of that, it's also duplicating its DNA. And the G2 phase is very similar to G1. It's still growing and doing normal life stuff. And cells spend most of their time in interphase. There's only a brief period of time where they enter mitosis and they actually divide up their genetic material and then split into two separate cells. So know that even though the bulk of this image is mitosis, know that interphase is actually taking up a lot more time in the cell's life history. Okay, but today we're going to focus in on what is happening in mitosis. Yes. So, in interphase, I said before, the cell is growing, doing its normal cell life kind of stuff. And if we focus in on what's happening in the nucleus, in interphase, the DNA is in its loose, uncompacted form. And this form is called chromatin. We'll talk more about the structure of chromatin, but know that it's DNA coiled around proteins, and together they form what's called chromatin. In interphase, the nucleus is intact, and the texture of the nucleus is kind of mostly uniform. Maybe you'll see darker and lighter splotches, but you mostly see a uniform mass or color here in the center of the cell. When the cell proceeds through interphase, there's going to be a point at which the DNA inside the nucleus is doubled. So now there's two copies of every chromosome. And the goal of mitosis is to divide those two chromosomes. So we ultimately get two cells that at the end are genetic clones. They exactly resemble each other in terms of the DNA that is inside of them. Okay, so how is this accomplished? Well, a few things need to happen in order to split up the DNA. So the cell enters into mitosis, and the first phase of mitosis is called prophase. In prophase, you'll start to notice that there are a few important things happening in the nucleus. First of all, the nuclear membrane begins to dissolve. And the chromosomes start to condense and form these X-like structures. And the last piece that you'll notice here is that this structure, which looks like little strings inside of the cell, this is called the spindle fiber. And the spindle fiber begin to form. The spindle fibers are pieces of the cytoskeleton. They're actually microtubules. We talked about them briefly at the beginning of the year, and they're going to be responsible for pulling apart the DNA, the chromosomes one from another. Okay, so the DNA in this condensed form, this X that it forms, these two pieces of DNA are genetically identical to one another. They're called sister chromatids, and they're all going to line up in the next phase of mitosis, which I'm going to give to you in a second, but I'm just going to shrink this down so these two things fit in the right spot. The next phase of mitosis is called metaphase. So, in metaphase, the chromosomes line up across this invisible equator of the cell called the metaphase plate. And like I just said before, these two arms of this one chromosome are genetic clones of one another. They were duplicated during S phase of interphase. So when they line up like this, we have all the information that the cell needs on this side of the equator, and then all the information that the cell needs in order to grow and live and survive on this side of the equator as well. So, one, chromosomes align on metaphase plate. The spindle fibers actually attach to the center of each chromosome here. It's important. It's a specialized structure called the kinetochore. And then the nuclear membrane is now entirely dissolved. Okay, so moving on to the next phase. This phase is called anaphase. At this point, you'll notice that the chromosomes have been cut. These two sister chromatids, these genetic clones, have been cut in the middle where the spindles were attached to them. And now they start to get pulled to opposite poles of the cell. So the spindle fiber pulls sister chromatids apart. So now we've taken the genetic information and we've split it in half. So now this side of the cell contains all of the DNA it needs, and this side of the cell also does as well. So, anaphase, you can actively see the chromosomes separating from one another, being pulled to opposite ends of the cell. And then the last phase of mitosis is called telophase, or some people say telophase. And you'll notice in telophase the nuclear membrane is starting to reform. The DNA starts to lose its characteristic chromosome-like shape and decondense into loose chromatin. And the spindle starts to dissolve. And in the center here this is a plant cell. You can tell because of its boxy-like appearance. It's got a cell wall around it. For a plant, this is called a cell plate. Starts to form from the interior of the cell. So this is both a cell wall and a cell membrane, or two cell membranes here, forming between what will be two distinct cells. In a plant, that starts in the center and it works its way out. And in an animal cell, it forms what's called a cleavage furrow. And so it starts from the outside and then works its way in. And we'll be looking at images of animal cells undergoing mitosis, so you'll see some of the structural differences between plants and animals there. And then this last piece, kind of telophase and cytokinesis are happening simultaneously. Telophase is really what is happening in the nucleus to the DNA, and cytokinesis is the splitting up of the rest of the cytoplasm. So remember, even though we can't see them in these pictures, there are a bunch of other organelles in here. Mitochondria and chloroplasts and rough ER and smooth ER. And all these things need to be divided between the two different cells. So that happens in cytokinesis, division of the cytoplasm and organelles. And then ultimately, we have two cells that are genetic clones of one another. And these independently can enter back into interphase, where they might go through these three phases again and divide, or they could enter the G not phase, where they are not dividing anymore. Okay, that's all. See you in class tomorrow.